Okay, Bismillahirrahmanirrahim. So our class today will continue on the process of neurulation and also organogenesis. Okay, just to recap a little bit, right? So our previous discussion is related to the process of gastrulation, right? So we have discussed the different gastrulation between frogs and also human, right? Okay, so the difference is due to the distribution of yolk. Okay, right. And at this point of stage, the embryo is known as gastrula, right? Okay, next, in the next stage following gastrulation is the process of neurulation. So what is neurulation? So neurulation is the process whereby the embryonic neural plate is formed and the transformation into neural tube. All right. So, and also by following neurulation is the process of organogenesis. So during organogenesis, the germ layers is differentiated into tissues and organ. So that is what we call as organogenesis, right? Okay. So organogenesis involves morphogenetic changes in the tissue and the cell shape, right? So at this stage, the embryo is known as neurula. Okay. So please inform me in case ada lagging ke apa ke bagi tahu madam. Okay. Right. So first it start with the process of formation of notochord and neural plate. Notochord and neural plate. Okay. Notochord form from dorsal mesoderm. Okay, remember mesoderm layer tu yang mana satu? Ectoderm is the outermost layer, mesoderm is the middle layer, and endoderm is the innermost layer, right? Okay, so notochord will be formed first. So notochord is formed from the dorsal mesoderm. What does it mean by dorsal mesoderm? It is mesoderm layer, tapi bahagian dorsal. Dorsal maksudnya? Bahagian? Atas, bawah, tepi, tengah?
Atas.
Atas, okay, dorsal mesoderm. So we have layer of mesoderm, right? So the dorsal mesoderm will condense. Okay, right. So notochord is formed from dorsal mesoderm that is condensed above the archenteron. Okay, let's look at this one first. Okay, you can see here, this is notochord. Okay, and this is what we call it? This is mesoderm from mesoderm layer. This is all mesoderm layer. So the dorsal mesoderm layer will condense, right? And it is dorsal mesoderm layer above the archenteron. So this is archenteron. So the dorsal mesoderm here is this one, right? So the dorsal mesoderm layer will condense and forming the notochord. Okay, right. So what is notochord? So notochord is flexible rod that will replace by vertebral column in vertebrates. So in other words, later on, the notochord will be forming ataupun will develop into the vertebral column, tulang belakang. Okay, so next, as notochord is formed, signal will be sent from notochord to the overlying ectoderm layer, meaning notochord will send signal to the layer above it. That is what we said as overlying ectoderm layer and cause that region of ectoderm to thicken and become neural plate. Okay, right. So look at this picture. Okay, so tadi ni kita dah tahu this is the notochord. Okay, we know that this is notochord. So notochord send signal to the above lying ectoderm layer. So actually this is whole ectoderm layer. So notochord send signal to the ectoderm layer above it or underlying ectoderm layer to thicken, as you can see from here, right? As it is thickened, it forms the neural plate. Okay, right? So this process of one tissue or one layer gives signal to another layer is normal, right? Where this process is often seen in organogenesis, where one germ layer signaling another to determine the fate of the next layer. So benda ni normal, one layer signaling another layer to do this and that, right? Okay. So once we have the notochord, we have the neural plate. Okay, next to be formed is neural tube, right? Okay, so this is our neural plate. Okay, next to be formed is the neural tube from the neural plate. Okay, okay. How the formation of neural tube happen? Okay, first, neural plate will invaginate. Neural plate will invaginate and forming neural groove. Okay, neural plate invaginate and forming neural groove. Okay, this one. Okay, neural plate invaginate like this and becoming neural groove. Okay, so ni groove lah. This is the groove. Right? Okay, next. Edge of the neural groove, called as neural fold, move towards each other and fuse. Right? So the edge of neural groove is called as neural fold. So the end of the neural fold fuse together and forming a long, hollow neural tube. Okay, this one. Okay. So this is what we call as the neural fold. Right? So the edge of the neural, bayangkan, the edge of the neural neural plate tadi forming neural groove. So as it form the groove, so hujung-hujung ni kita panggil sebagai neural fold. So the edge of the neural fold meet together. Dia bersambung, hujung-hujung tu bersambung, right? This one and this one meet at the end, right? To form neural tube. Okay? Right. Okay, tengok ni tadi. Okay, so edge of neural tube, which is known as neural fold, move towards each other, fuse, and forming long, hollow neural tube. So the neural tube is located beneath the surface of the embryo's back, right? So the infolding and pinching of the neural plate will generate the neural tube. So as they fuse or pinch off, then it will form neural tube. Okay. What will happen to the neural tube? The neural tube differentiate into brain and the spinal cord. Okay, so we have notochord yang akan jadi tulang belakang, right? And then neural tube differentiate into brain and the spinal cord. Okay, right, like this one. Okay, so again, this is the neural groove and this is the neural fold that deeper, okay, become fused together. You can see, right? Which later on they will fuse. But just before the neural groove closes or fuse, some cell from the dorsal margin, some cell yang dari atas ni, right? Some cell of dorsal margin of the neural tube differentiate into neural crest. Neural crest then migrate to many parts of embryo. So this is neural crest. And this is the neural tube that has been formed by fusion of the neural fold tadi tu. Right, maksudnya sebelum neural fold tu tutup, bersambung, some cell of the dorsal margin, maksudnya some cell daripada atas akan membentuk neural crest. What will happen to the neural crest? Neural crest will develop into gill arches, okay, ataupun macam parts of macam insang, something like that, for respiration. Sensory ganglia, Schwann cell, periphery nerve, teeth, okay, skull, bones, and also adrenal medulla. So this is developed from neural crest. Okay, right? Contoh macam ni. This is the picture just now. Okay, this one. So you can see before the neural tube fuse or neural groove fuse, right? You can see that some cell of the dorsal margin will form neural crest. So dia akan duduk dekat bahagian atas macam ni. So that is neural crest, and here you have the neural tube. Okay, right. So that is how notochord, neural tube, and neural crest is formed. Okay. Any question? Any question?
No, madam.
Okay.
All good.
All right. So again, you have here tadi. This is the neural groove that form the neural fold. So what happened when before the neural fold closes, then some cell of the dorsal margin follow the neural crest. Okay. All right. Okay, so finish with the development of the nervous system. So we finish with the process of neurulation. So at this stage, the embryo is known as neurula. Okay, so you have to, focus and bear in mind or remember that, let's say, kalau question keluar, soalan keluar, and given to you a picture of stages of embryonic development, right? So if you see the picture of neural tube, the notochord, right? Development of neural crest. So that is actually referring to the process of neurulation. So the embryo is said as or in the stage of neurula. Okay. All right. Next, we go to the the whole process of early embryonic development of human. All right, so why we know that this embryo is human embryo? What makes we know that this is human embryo?
Trophoblast.
Yes, you can see because the structure that have the trophoblast cell, right? The inner cell mass, okay? Where this is what we call as the human blastocyst, okay? Right. Okay, so the whole process of embryonic development. So remember, fertilization start with the process of fertilization, where the fertilization happens in the fallopian tube, or we call as oviduct, right? So the major stages of early development of human involving the blastocyst reach to the uterus, right? So maksudnya the developmental start, right? Secara orang kata apa, major as blastocyst reach to the uterus and the blastocyst implant in the wall of uterus, right? And after then, gastrulation and formation of extra embryonic membrane began, right? So what is extra embryonic membrane? So extra embryonic membrane, main membrane, basically 4 membranes, right? That assist in the development of embryo, right? What are the extra embryonic membrane? So we will discuss afterwards. Okay, so next, after the fertilization in oviduct, so we know that embryo will implant in the uterus, okay? But before that, cleavage occur throughout the movement of embryo within the fallopian tube into the uterus. So cleavage occur as embryo migrates to the uterus through oviduct. So how embryo can migrate within the oviduct or fallopian tube? That is assisted by the peristaltic contraction of the oviduct, right? It is smooth muscle, right? And then the sweeping ciliary motion, as we can see, the lining of fallopian tube is lined with cilia, ciliated epithelium. So the ciliary movement, right, of the cilia helps in the movement of the egg or the embryo. Okay. Right. Okay, implantation process. Okay, so remember I did mention in class that the implantation process happen maybe around 7 to 8 days, right? Or up to the process altogether, maybe up to 12 days after fertilization. So usually around 7 to 8 days. Okay, blastocyst implanted in the wall of uterus, right? Around 7 days after fertilization. So 8 days onward, trophoblast implant into the endometrium and become chorionic membrane. Okay, trophoblast implant. What does it mean by trophoblast implant? So we have here, this is the trophoblast. Ingat balik kan? Ini trophoblast. Ini inner cell mass that has rearranged into epiblast and hypoblast. So this is trophoblast. You can see that trophoblast implant in the wall of endometrium and become chorionic membrane. Okay, what does it mean by chorionic membrane? What will happen to chorionic membrane? So later on, chorionic membrane will develop into placenta. Okay, right. Okay, next. Okay, so after the implantation, okay, inner cell mass rearrange and forming amniotic cavity and disc of cell below the cavity, right? So kalau ikut tadi kita tengok inner cell mass rearrange into hypoblast and epiblast je kan? So now we become, kita detail out a little bit. So inner cell mass rearrange and forming amniotic cavity. Which one is amniotic cavity? This one. Okay, ini amniotic cavity. Alright, and disc of cell below the cavity, yaitu epiblast and hypoblast. Alright, so how to again differentiate which one is epiblast, which one is hypoblast? Kita discuss dalam class. Hypoblast is the one that very close to the siapa nama tu? Blastocyst, right? And epiblast is near to the amniotic cavity. Okay, so macam kalau dalam gambar tu kita akan nampak macam ni. This is epiblast. A dome-like structure here. Cavity, amniotic cavity. So this is epiblast. And here is hypoblast. And then yang kat sini tu, malas nak lukis. Ni semua ni. So ini adalah blastocyst. Okay, so this is hypoblast. Okay, all right. So the disarrange to form epiblast, okay, and also hypoblast, right? So gastrulation begin when the cell of epiblast, okay, migrate inwards or invaginate inwards through the primitive streak, right, and forming mesoderm and endoderm. All right, so this is where gastrulation started. When the cell of epiblast migrate or invaginate inwards through primitive streak. Remember, kita dah discuss tadi, some epiblast cell, okay, migrate sideways ataupun move sideways or laterally and forming mesoderm. Some epiblast cell move downwards, displacing
hypoblast.
Yes, hypoblast cell displacing hypoblast and becoming endoderm. And the epiblast cell that remain on the surface will become ectoderm layer. All right, okay. And then after the formation of the 3 germ layers, then extra embryonic membrane will start to form. All right. So what are the extra embryonic membrane? Okay. So these are the extra embryonic membrane. They are, okay, amnion. Okay, yolk sac, right? And another one here is allantois. Okay, so this is chorion. You see, chorion is the outermost layer surrounding the embryo, right? And that is chorion. The blue color is the amnion, okay, derived from the ectoderm layer, right? And then the yellow color here is the yolk sac, right? And then some structure that protruded, but this is not fully developed yet. So this is allantois, right? So this happened around 16 days after fertilization. Okay, so these are the 4 extra embryonic membrane. Okay, let's discuss. Okay, ni tadi yang Madam cakap tu. Ni epiblast, this is epiblast, this is amniotic cavity, right? And down here is the hypoblast, and this is the blastocyst initially, which later on will develop into yolk sac. Okay. Okay. Next one. Developing offspring. Okay. The first 8 weeks ataupun the first 2 months of pregnancy, right? The baby ataupun baby tu kita panggil sebagai embryo because they are still develop crucial ataupun essential tissues and organs. Okay. And then the 3rd month onwards, right? The baby will be known as fetus. Right, so that's why people say the first trimester tu, right? First trimester is 3 months. Setiap trimester ialah 3 bulan. So we have a first trimester is the first week into the 3rd week, the 3rd week of pregnancy, right? Apa pula ni? 12 weeks of pregnancy. Okay, the first week sehingga berumur 12 minggu. 1 minggu, 1 bulan ada 4 minggu. 1 bulan ada 4 minggu. So 3 bulan ada 12 minggu. So the first 12 week of pregnancy is crucial, right? For the developmental stage of embryo. Okay. So the developmental developmental stage of embryo, or we call as gestational stage. Tempoh kehamilan. Gestation stage ialah tempoh kehamilan. Okay, gestation stage for human. Okay, I'm talking about human, bukan gajah ke apa ke. Okay, the gestation stage of human is around 266 days, which divided into 3 trimesters. Okay, so 266 hari itu lebih kurang dalam 14 weeks. Okay, right? So each trimester is equal to plus minus 3 months. Okay, right. So you can see the first 8 weeks. Okay, the baby is known as embryo. And the last 7 months, right? So the baby is known as fetus. So this is the range of 14 weeks of pregnancy of human. Okay, right. So in Quran, Allah did mention about the developmental stage of human, right? In Surah Al-Mu'minun, first is verse 14, right? So you can see that Allah mentioned here, Summa alaqatan nutfatan alaqah. All right. So then of that fluid drop, the nutfah. Nutfah, we created a leech-like clot. So nutfah become alaqah. Right? So alaqah is leech-like clot ataupun bahasa biasa kita panggil macam segumpal darah. Okay, right. So tu, yeah, betul. Segumpal darah. Okay. So the transformation process from nutfah, right, from the fluid drop to alaqah takes more than 10 days. So kita pun dah belajar tadi. So the process of implantation takes around 7 to 8 or 7 to 10 days. Okay, of post-fertilization. Okay, until the zygote cling to the primitive placenta by way of connecting sock, which later become umbilical cord. Alright, so Quran use conjunction summa. Kenapa summa? Right, the summa is actually indicate sequence of event with a time delay instead of using fa. Fa means kemudian. Kemudian tu menunjukkan dia berlaku dengan cepat. So kat sini nak bagi tahu yang ayat Quran tu guna pun is actually related to whatever mentioned in ataupun whatever that we discover in science, where from nutfah to alaqah it takes around 8 to 10 days untuk jadi. So summa means the process berlaku selepas beberapa lama. So next, okay, in the next ayat, right? Fa khalaqnal alaqata mudghah. Then we change alaqah into mudghah, seketul daging. Right, a quick transformation then occur from alaqah to mudghah. That happened within 2 days, day 24 to day 26 of fertilization, after fertilization. Maksudnya proses dia cepat, daripada segumpal darah kepada seketul daging berlaku dalam tempoh masa yang singkat, berbanding dengan daripada nutfah kepada alaqah. That's why the Quran use conjunction fa instead of summa. Nampak tak kat situ? So benda ni kita nak kata apa? Relate whatever that we learn in science to Quran. Okay, next, the stage of bone and muscle. So nutfah, alaqah, mudghah. Next, fa khalaqnal mudghata izama. Right, then we made out that mudghah, bones, and clothe the bones with flesh. Right, so this is how the human embryonic developed based on whatever mentioned in Quran, which later we found in science. Okay, right. Okay, now let's go and discuss about the extra embryonic membrane. Okay, so we will see the role of chorion, amnion, yolk sac, allantois, and placenta. So you might be wondering, madam tadi bila kata 4 kan? Sebab apa? Sebab chorion dengan placenta ni somehow related. Okay, so itulah. Okay, am I to first? Can you follow?
Boleh.
Boleh, okay. Okay, next. Okay, now let's see. Okay, this one is important. So you have to make sure you have this picture of embryo with all the extra embryonic membrane. Okay, so first chorion. This is chorion. So ingat chorion yang lapisan yang paling luar sekali. This is chorion. Okay, so chorion plus the other villi. Okay, what is villi? Okay, later I'll explain. Okay, next one. Another layer is amnion. So this is amnion. Okay, this is amnion layer. And this is the embryo. Okay, developing embryo. Right, next, inside the amnion there is fluid or cavity known as amniotic cavity. All right, and here we have yolk sac. This is yolk sac, right? And this is allantois. Okay, this is allantois. All right, so this is the first picture, right? Okay, now let's go into the detail of each extra embryonic membrane. Okay, first, chorion. Okay, so chorion surround the embryo and all the extra embryonic membrane. So meaning to say chorion is the outermost layer, right? Sebab dia yang meliputi the embryo and all the other extra embryonic membrane. Okay, so this is chorion. Okay. Right, okay. Chorion form finger-like projection known as chorionic villi. So this is the villi that you see. Okay, this is what we call as chorionic villi, right? That will project and penetrate deep into the endometrium tissue. Okay. Right. So these chorionic villi project into the blood spaces in the uterine wall. Okay. Chorion will become chorionic frondosum, which is the fetal placenta, right? And surrounding the epithelial tissue is decidua basilis. Decidua basilis is maternal placenta. Okay, do I have a picture? Okay, this one. Okay, so again, this is chorion. Okay, you can see chorion is the outermost layer that surrounds the embryo and all the other extramembrane membrane, right? So it forms projections into the wall of uterus, right? And then chorion form chorionic frondosum. Which one is chorionic frondosum? This one. Okay. And decidua, sorry. Chorionic frondosum yang mana satu? Ini macam bunga-bunga ni. This is chorionic frondosum. Okay. And decidua basilis yang kat luar ni. Okay, that is decidua basilis. Okay. Chorionic frondosum and decidua basilis. Okay, tengok balik ayat tadi. Chorion, okay, chorion become chorionic frondosum, which is the fetal placenta. Surrounding the epithelial tissue, meaning surrounding the chorionic frondosum is decidua basilis. Decidua basilis is the maternal placenta. So together, chorionic frondosum and decidua basilis will form the placenta. Placenta tu apa? Placenta tu apa? Bahasa Melayu kita panggil apa?
Uri.
Uri. Okay. Placenta ialah uri. Okay. Bukan tali pusat tali pusat umbilical cord. Okay. So dia akan jadi placenta atau kita panggil uri. Okay. Right. So you can see that the mother's and the embryo blood is very close together. This is the embryo blood, right? And this is the mother's blood. So they are very close together, but they are not mixed. Okay, itulah function placenta lah. So placenta has a very huge function for both benefit of the mother as well as the fetus. Okay, so what are the function of placenta? So first, placenta help in assisting gases exchange, where oxygen from the mother can reach the embryo and carbon dioxide in the opposite direction. Okay, number two, placenta provide nourishment for the embryo. So nutrient from the mother can pass to the embryo via placenta. Okay, next, placenta also helps to detox certain molecule that pass into the embryo. So it somehow helps to protect the embryo from harm. Contoh kalau mak tu ambil dadah ke, mak tu ambil alkohol ke, right? So somehow placenta can help to detox certain molecule that pass into the embryo. Okay, right. Next, placenta also replace the role of trophoblast to secrete hormone HCG that detects pregnancy. Okay, maknanya apa? In the early pregnancy, right, HCG is released by trophoblast. Masa early pregnancy. Macam you baru seminggu delay period ke, 2 minggu delay period ke, so macam mana hormon HCG itu keluar ataupun dihasilkan? It is secreted from the trophoblast. But as the embryo develop, okay, grows, jadi makin besar, makin besar, makin besar. So the role of secreting HCG will be taken by the placenta as placenta develop. Okay, right. So what is the function of placenta? Okay, what is the function of HCG? So HCG maintain the corpus luteum, right, by secreting estrogen and progesterone, which help to thicken the endometrium wall to support pregnancy, as well as to prevent maturation or ovulation, right? So HCG helps to maintain the corpus luteum so that estrogen and progesterone can be secreted. Okay. Okay, so that is function of placenta. Okay, ada soalan tanya je ya. Okay, next one is amnion. Okay, amnion is the innermost membrane. Innermost membrane. Okay, so kita tengok balik gambar yang ni. Do we have another picture? Okay, ada. Right. So amnion is the innermost membrane. It forms as a dome. Bentuk dia macam dome, right? Macam kubah above the epiblast. Alright. Okay, kita tengok sini. You can see here. This is epiblast. Ni yang warna biru ni. This is epiblast. So it forms as a dome. Ni. Bentuk dia macam kubah above the epiblast, right? So that is amnion. Okay, it form a dome as a dome above epiblast. Okay, kalau tengok gambar yang ada baby, okay, mana satu dia? So this is amnion. Okay, next layer. That is amnion. Right, so chorion, lepas tu next layer is amnion. Okay, right. Okay, amnion lines the fluid amniotic cavity. Amnion lines the fluid filled amniotic cavity. So dalam amnion tu ada cavity. We call as amniotic cavity. What is the function of the amniotic cavity? Right, dia ada amnion fluid, right, that encloses amniotic fluid that enclose and cushion the embryo in the fluid filled cavity, right? The fluid also prevent the embryo from drying out and permit freedom of motion. Okay, so kalau nak jadikan cerita, amnion tu apa? Kalau placenta tu uri, amnion tu apa?
Ketuban.
Apa?
Ketuban.
Dia adalah kantung. Dalam kantung tu ada cavity. Dalam cavity tu ada cecair amniotic fluid, which kita panggil sebagai air ketuban. So the amniotic fluid tu ialah air ketuban, right, within the sac. And the sac is the amnion lah. Okay, kantung. Kita panggil sebagai kantung. Okay. Maknanya kalau wanita bersalin, yang akan keluar tu uri sekali dengan kantung tu. Kantung tu ada dalam tu kalau kita tengok. Okay, right. So the function is to cushion the baby, okay, from shock, right? It also helps to protect the baby from, or protect the embryo or fetus from shock, ataupun in other words we say as to cushion the embryo, okay, and prevent the embryo from drying out. That is amnion. All right. Okay. Next we go to the third one, yolk sac. Okay, yolk sac. Okay, for human, right? There is a sac ataupun sarung ataupun kantung, right? Juga, this is yolk sac, right? You can have this picture. This is yolk sac in chick, ayam punya, and this is human punya. For human, the yolk sac is enclosed with filled cavity, but no yolk. Okay, for human, kita ada cavity tu, sarung tu ada, kantung tu ada, tapi tak ada yolk. Kalau untuk ayam ke apa ke, dia ada yolk dia tu. Okay, right. So, no yolk for human. So, what is the function of yolk sac? So, yolk sac serve as temporary site for early formation of blood cell before the bone marrow develop, right? So, sel darah semua dihasilkan dekat yolk sac, right? And yolk sac will diminish as placenta develop and expand. Okay, so apabila placenta dah makin membesar, okay, uri itu dah makin membesar, right? Then yolk sac will diminishes. So antara satu benda or indication for detection of early pregnancy, doktor akan nampak yolk sac. Okay, so bila scan tu nampak ada kantung tu, itu antaranya kita panggil sebagai yolk sac. Okay, right. So this is the yolk sac. Okay, right. Okay, next one is allantois. Okay, next one is allantois. Okay, allantois is very small. Okay, allantois is very small and non-functional. So where is allantois? This is allantois. Okay, you can see somehow allantois is look somehow it is connected to chorion. Allantois is very small and non-functional. It developed from outgrowth of the developing digestive tract. So dia seolah-olah develop macam sesuatu benda yang terkeluar, macam satu benda yang terkeluar ataupun tertonjol from the development of digestive tract. Okay, so macam inilah, macam dia terkeluar daripada development of digestive tract macam tu. Okay, allantois is incorporated into umbilical cord where it forms the blood vessel. Okay, tu salur-salur darah is developed from umbilical cord, alright, where it form the umbilical artery and also vein, atau bahasa Melayu kita panggil tali pusat. That's why tali pusat tu macam seolah-olah keluar daripada sebahagian daripada kita punya digestive tract. Keluar daripada pusat baby tu kan. Alright, so it somehow looks like an outgrowth from the developing digestive tract. Okay, alright. Function of umbilical artery and vein, right? So the function of umbilical artery and vein is to transfer oxygen and nutrient from the placenta to embryo and waste from the embryo to placenta. That's why you can see that somehow allantois is related to chorion or related to the placenta. Okay. Right. Okay, any question? Ada?
Madam.
No, madam.
Okay, right. Kalau tak ada, Madam sambung. Okay, the process of parturition and lactation. Okay, so our LO is to describe role of hormone, the role of hormone during labor, right? Parturition is the action of labor, and lactation. Lactation ialah penyusuan. Okay, what is parturition? Okay, parturition is the act of giving birth ataupun the process of labor. Okay, right. So, tengok balik. Ni apa ni? Apa tu? Which layer extra embryonic membrane is this?
Amnion.
This is amnion. Right? So you can see it lines the amniotic cavity, right? Which filled with amniotic fluid, right? And here is part of the chorion that form the placenta, and here is portion of the allantois that develop into the umbilical cord. Okay. Okay. So stage of labor. There are three main stages of labor. The first stage of labor is dilation of cervix. Cervix akan mengembang, right? So you can see here, this is actually opening of vagina. Ini vagina ya, right? And this is cervix. You just imagine how far has the cervix dilated, right? So near to the process of labor. Okay, so the dilation of cervix takes around 12 hours and can be up to 24 hours to dilate in the first pregnancy, or it can be just few minutes after many pregnancies for the cervix to dilate. What happened to the cervix? The cervix become thin and expand. Then the baby's head will pass from the cervix into vagina. You can see that the baby's head, part of the baby's head is already here, right? Dah masuk dekat sini. So kalau kita tengok dari sini, view from here, ni mata, right? Okay, so we can actually feel and see the baby's head already. Okay, so the baby's head pass into the vagina, amniotic fluid will release from the uterus. Ataupun dalam kata lain, air ketuban pun dah pecah. Okay, by the rupturing of amnion. Okay, cervix dilate up to maximum of 10 cm. Okay, bila dah share dalam kelas kan, macam mana nak check cervix kan? Using fingers. Okay, right. So that is the first stage of labor. So as the cervix dilated, dah masuk 10 cm, then we go to the next stage, which is expulsion or delivery of infant. So ni cervix tadi. Right, this is vagina. So you can see what happened. Right, okay. So the delivery of infant takes around 20 to 60 minutes, right? Where uterine contraction becomes stronger, uterus contract, right? And become more frequent. Makin kerap dia punya contraction. And then the baby will be pushed through the vagina and then out of the mother's body. Okay, right. Okay, next, third stage. Third stage of labor is delivery of placenta, or we call as the afterbirth, right? So placenta will be delivered 10 to 15 minutes after the baby is born, right? Where another contraction or another series of contraction will happen, right? Further contraction of uterus will happen and push umbilical cord and placenta out of the mother's body. So you can see this is the ejected placenta and part of the umbilical cord. All right, and you can see that this is the cervix, right? Cervix will be, orang kata apa, mengecut sedikit, right? And you can see that uterus is now empty. Okay, so this happened 10 to 15 minutes after birth. Okay. Soalan dia sekarang ni ialah, how hormone initiate or induce the process of labor, right? Macam mana hormon mempengaruhi ataupun mengambil tanggungjawab, right, during parturition. Okay, that is the question. Okay, right. So this is the summary of the first stage of labor, right? You can see this is when the baby is fully developed fetus, right? And then belum lagi sampai tahap cervix open, right? So this is what happened the first stage of labor, dilation stage. Okay, ingat kalau nak tahu dilation stage, maybe mesti kepala baby dah lalu dekat cervix, dia dah duduk dekat hujung-hujung vagina. So that is the dilation stage. Right? So expulsion stage, the baby is kata apa, pushed out from the mother's body, right? And this is the next stage, which is the delivery of placenta. Okay. Okay, now how hormone involved in the process of parturition? Okay, so in the last week of pregnancy, hujung minggu-minggu nak bersalin tu, minggu 38, 39 semua tu, alright, there will be high level of estrogen. Okay, high level of estrogen during the last weeks of pregnancy. So the high estrogen level trigger formation of oxytocin receptor on uterus. So high estrogen induce the formation of oxytocin receptor on uterus. Okay, right? So remember what is oxytocin? Right, oxytocin is one of the hormone released by the pituitary gland, posterior pituitary gland, and it is released from both the fetus and the mothers, or in the maternal posterior pituitary gland. Maknanya dua-dua, mak dengan baby, dua-dua keluarkan oxytocin. Okay, so what is the function of oxytocin? Oxytocin stimulate the smooth muscle of uterus to contract via positive feedback. Maknanya oxytocin contract, smooth muscle, oxytocin release, smooth muscle contract. Lagi kuat contraction, lagi banyak oxytocin yang keluar. So it is positive feedback. Okay. So oxytocin stimulate the smooth muscle of uterus to contract via positive feedback. Okay. Next, what happened? Okay. Oxytocin also, ingat so the first function of oxytocin tadi to contract the uterus, right? Via positive feedback. Second function of oxytocin is to stimulate the secretion of prostaglandin by placenta. Stimulate the secretion of prostaglandin by placenta. So apa tugas prostaglandin? Tugas prostaglandin is to enhance. The word enhance must be there. Okay, mesti ada ni. Right? Prostaglandin enhance muscle contraction. So bila contraction mak sakit. Lagi mak sakit, lagi banyak prostaglandin keluar, lagi kuat contraction of muscle. Contraction of uterus. Okay, right. The physical and emotional stress caused by the uterus contraction stimulate additional oxytocin and prostaglandin. Lagi mak sakit, lagi banyak oxytocin, lagi banyak prostaglandin. So it is positive feedback, right? So more contraction of the uterus, so baby can be safely delivered. Okay, right. And after the baby has been safely delivered, so after birth, the level of progesterone will drop, right? Actually, together with estrogen, okay, both estrogen and progesterone will drop to allow the production of another hormone, which is prolactin. What is the function of prolactin? What is the function of prolactin?
Penyusuan madam.
Apa penyusuan? Apa yang berkaitan dengan penyusuan?
Penghasilan susu.
Yes, okay. So prolactin induce milk production. All right, so prolactin induce milk production. Okay, right. So after birth, level of progesterone will low, as well as the level of estrogen, which then induce another hormone to be in action, which is, for example, prolactin. Okay. Okay, so you can see from here, right? At the late weeks of pregnancy, okay, the level of estrogen increases. Tadilah to induce the oxytocin receptor. In the early week of pregnancy, we can see that HCG is very high to detect the presence of pregnancy, to thicken the endometrium wall, kan? And after that, you can see progesterone start to increase. Ni masa throughout the pregnancy, right? But as you are finished with parturition, bersalin je, estrogen level will drop, okay? Progesterone level also will drop. Okay, right. So kita nak fokus kat mana? Kita nak fokus sini. At the late week of pregnancy, estrogen level is increased. Okay. Okay. So this is the picture to summarize the process of labor, right? So remember, at the late pregnancy, estrogen level will be higher, right? So estrogen will be travel from the ovary and then act to the what we call it, target cell. Target cell dia adalah, one of it is the uterus. It will induce the formation of oxytocin receptor on the uterus, right? So oxytocin then released by both the fetus and the mother's posterior pituitary gland, where oxytocin stimulate the contraction of uterus. Okay, so the contraction of uterus also stimulating placenta to release or to secrete hormone prostaglandin. So the function of prostaglandin is to enhance contraction of uterus via positive feedback. So more contraction of uterus happen. So the more contraction of uterus, more placenta will be released, as well as more oxytocin until the baby is delivered. Okay. Okay. So Allah mentioned in Surah Al-Hajj, okay, verse number 5, right? And we cause whom we will rest in the womb for an appointed term. Right, so kat sini nak bagi tahu yang Allah bagi menceritakan tentang masa, right? Tempoh kehamilan, for example. So ada orang yang Allah takdirkan, the baby will be safe in the womb sampai 9 months. Ada yang Allah takdirkan 7 bulan, 8 bulan dah bersalin. Ada yang sekadar 2-3 months and then miscarriage. So Allah yang mengizinkan benda itu berlaku. So we will cause whom we will to rest in the womb for an appointed term. Right? So after the passing 9 lunar months, the fetus is complete in terms of growth in the uterus. Right? And then here in Surah Al-Mursalat, the term is appointed and determined. Right? So kat sini, the which fetus we place in the place of rest, which is womb, firmly fixed for a period that is determined. Pada satu-satu masa yang telah ditetapkan. And we are the best determiner. Right? Itulah. Okay, so next. Baby is safely delivered. So the mother will continue with the process of lactation. Okay. So lactation. Okay, kita habiskan lactation ni and then kita break for 5 minutes and then we proceed with plant development. Okay? Right. Okay, lactation. What is lactation? Lactation is the process where ataupun bahasa Melayu kita panggil proses penyusuan. Okay? So secretion of milk from the mammary gland, okay, where the secretion of milk from mammary gland involve 2 hormones. They are prolactin to promote milk production, okay, and also oxytocin. Oxytocin stimulate ejection of milk from the mother's nipple, right? So maksudnya these 2 hormones must be present. Kalau prolactin ada, oxytocin tak ada, pun tak guna. Maksudnya the milk is produced, but the milk cannot be ejected. Susu tak boleh keluar. Susu ada tapi susu tak boleh keluar. Right? So both hormones must be present, prolactin and oxytocin. Okay? Right. Prolactin is secreted by anterior pituitary gland. Oxytocin is secreted from the posterior pituitary gland. Okay? Right. So the effect of prolactin and oxytocin is inhibited by progesterone and estrogen. Okay, that's why. Okay, maksudnya apa? After birth, the level of progesterone and estrogen. No, after birth. Right, so the low level of progesterone and estrogen will induce the increase of prolactin and oxytocin. Right, but let's say, right, dikata tadi the effect is inhibited by progesterone and estrogen. Let's say progesterone and estrogen increase, so dia akan inhibit pula prolactin and prolactin and oxytocin. That's why kalau ibu mengandung, right, bila mengandung progesteron akan tinggi, estrogen akan tinggi. Somehow dia akan affect prolactin and oxytocin. So dia akan affect the production of milk. Okay, macam itulah lebih kurang. Okay. Right, after birth, the placenta is discharged. So the level of progesterone and estrogen will fall. Okay, so the declining of progesterone and estrogen will enable prolactin to promote milk production. Okay, prolactin level increase temporarily every time the baby suckles from the mother. So maksudnya lagi banyak baby menyusu dengan ibu, prolactin level will increase. So more milk will be produced. So it's demand versus supply. Okay. Thus the milk supply is regulated to the demand of the baby. So the more baby feeds, the greater the prolactin secretion and milk production. And this is again showing the positive feedback. Sebab tu kalau ibu bekerja ke apa ke, dia akan still try to pump the milk, right, to induce more prolactin. So more milk will be produced. Okay, macam itulah. Okay, suckling also, as the baby feed from the mother, right, we call as suckling. So when the baby suckled from the mother, it also trigger the release of oxytocin. Ingat tadi apa function oxytocin in this process of lactation? So oxytocin ensures milk can flow freely. Untuk pastikan susu boleh mengalir. Susu ada dan susu kena mengalir dengan baik. So that is the function of oxytocin, ensures milk flow freely. Okay, so the suckling of baby also, despite trigger the release of oxytocin, the suckling also inhibits secretion of LH and FSH, whereby, remember, LH is luteinizing hormone, induce the process of ovulation. FSH, follicle stimulating hormone, right, induce the development of follicle. So when as the baby feed from the mother, it can actually inhibit the level of LH and FSH. So the process is called as lactational and ovulation. In other words, kalau selagi ibu itu menyusukan anak exclusively, right, breastfeeding exclusively ataupun pump susu ke whatever ke, so it will reduce LH and FSH. In other words, no ovulation, right, no development of follicle, so it can actually orang kata apa, kita panggil reduce chances of pregnancy. That's why Nabi kata susukan anak sampai 2 tahun. Sebab bila kita susu exclusively anak tu 2 tahun, so the mother will actually not having their menses. Dia takkan period sebab no level of LH, no level of FSH, right? No development of follicle, tak ada ovulation, so no chances of getting pregnant, and also mungkin juga dia takkan period. This is based on science, but there are of course a few cases, despite my own friend yang exclusively breastfeeding her child still pregnant, right? Walaupun dia tak period, tapi dia pregnant. So that is something that away from science can explain. Okay. But when nursing stop, so bila ibu tu stop menyusukan, accumulation of milk in the breast will signal the brain to stop the production of or the secretion of prolactin. So milk production will be ceased. Okay. So that is lactation. Okay. Any question? Any question?
No, madam.
No. Okay. So kalau tak ada soalan, we take 5 minutes break and we will continue after this with plant development. Okay. Right. So take 5 minutes. Right. Next, let's go to the plant development. Okay. Right. So the LO for plant development includes the describe development of seed and fruit. Development of seed and fruit. Okay. Right. So we start with the seed development. Okay. Under the development of seed ada dua. Right. Satu, development of the endosperm, and number 2 is the development of the embryo. Okay. So now let's discuss the first part, which is the development of endosperm. Ingat balik function endosperm tu apa? Ingat tak? What is the function of endosperm? To
Provide nutrient.
To?
Provide nutrient.
Yes, provide nutrient for the developing embryo. Okay. So endosperm is developed before the embryo. Maksudnya, siapa nama tu? Endosperm will develop first, then the embryo develop. Sebab it is source of nutrient or to support the development of embryo, kan? Right. So how does endosperm form? Ingat balik, endosperm form the triploid nucleus, right? That form and divide repeatedly, forming multinucleated cell, or we call as supercell of milky consistency. Okay, so this is example of corn canal, right? So this is the endosperm. So ini semua adalah kawasan untuk the endosperm. So this is the cross section. Okay, so endosperm develop first before the embryo. Okay, so it is formed from the triploid nucleus, right? And the function is to provide nutrition and support for the embryo. All right. Okay, and then what happened to the endosperm? Endosperm will become solid. Okay, right. Okay, next. What are the nutrients needed for the developing embryo? So the developing embryo needs starch, protein, and lipid. So they are stored in the endosperm for corn, for example, because corn is monocotyledon plant, right? And for eudicote, like common garden bee, for eudicote, the nutrient is stored in cotyledons. Okay, so for monocot, right, the starch, protein, and lipids stored in endosperm. For eudicote, like garden bean, it is stored in cotyledon. Okay, in grains, most monocot and many eudicote, the endosperm stored nutrient that can be used by the seedling after germination. Okay. But in other eudicote seed, nutrient from the endosperm will be exported from endosperm to the cotyledon before the seed complete its development. And thus, mature seed is lacking of endosperm. Okay, dia nak cerita apa kat sini? In grains. Grains apa grains asal kita nak panggil apa hilang perkataan grains ni? Macam gandum ke apa ke semua tu kan, right? So in grains, most monocot and many eudicote, usually the endosperm will store the nutrient and used by the seedling after selepas germination. But for other eudicote, okay, nutrient from endosperm will be transported to cotyledon. Okay, contoh kita tengok kat sini. This is bean, right? Example of eudicote. And this is corn canal, right? Example of monocot, right? For monocot, usually the nutrient is stored inside the endosperm. Okay, but for many eudicote, like other seeds like beans, for example, right? So the nutrient will be stored inside the cotyledon. Okay, so the nutrient stored in the cotyledon. Itu je lah dia nak bagi tahu. And thus mature seed is lacking of endosperm because they have cotyledon. Okay, next is that is endosperm development. Next is embryo development, right? So you can see from here, and this is zygote, right? Zygote will then form terminal cell and basal cell. Okay, let's see how the zygote form terminal cell and basal cell. Okay, so this is the whole thing actually showing you the development of embryo. Okay, right, kita start satu-satu. Okay, first, right? Okay, so first, this is the zygote. The zygote will undergo asymmetric division. Okay, asymmetric division means pembahagian sel yang tak sama besar, right? Which will form a smaller cell. Ni small cell ni known as terminal cell, and larger cell down here is the basal cell. So due to the asymmetrical division, it forms smaller cell and bigger cell. So the smaller cell is known as terminal cell. The bigger cell is known as basal cell. Okay, what happened to the basal cell? Basal, okay, base. Okay, what happened to the basal cell? Basal cell continue dividing, right, and forming a row of cells that we call as suspensor. So basal cell continue dividing and forming suspensor. So this is suspensor. Alright. Okay, what is the function of suspensor? Maknanya basal cell ni divide 2 cell, 4 cell and so on. So jadilah dia suspensor. Okay, what is the function of suspensor? Suspensor transfer nutrient from the endosperm or from the parent plant to the embryo. Okay, mana datang embryo ni? Embryo ni datang daripada terminal cell. So suspensor provide nutrient to the embryo. Okay. Right, what happened to the terminal cell? Okay, yang ini ni apa jadi dengan terminal cell ni? Yang ni tadi. So terminal cell divide repeatedly and forming a globular or spherical pro-embryo. So they divide. Jadilah dia ni. Spherical or globular pro-embryo. So this is pro-embryo. Okay, the pro-embryo is attached to the suspensor. Pro-embryo attached to suspensor. Right? Okay, next. Early formation of cotyledon. Ingat cotyledon tu maksudnya apa? Cotyledon ni sebenarnya adalah embryo. Okay, right. Early formation of cotyledon in dicot or in eudicote give the embryo a heart-shaped appearance. So this is the initial appearance of the embryo. So ni heart-shaped. Okay, heart-shaped embryo. Then the embryo elongates and changes into a torpedo shape. So this is torpedo shape embryo yang kita panggil sebagai cotyledon. Okay. So that is how embryo is developed, right? Okay. So kita tengok balik kat sini. Okay. So again, this is terminal cell, this is basal cell. Basal cell divide and forming suspensor, right? Terminal cell will develop into pro-embryo. So this is the heart-shaped embryo. This is torpedo shape embryo, but still attached to the suspensor. And later on, you can see it form this one. This is the embryo, the cotyledon. Inilah dia, cotyledon. This is embryo, and this is the suspensor. Nampak tak? So suspensor transfer nutrient from endosperm daripada sini hantar ke embryo. So that is the function of suspensor. Okay. Okay. Structure of a mature seed. Okay. Okay, structure of a mature seed. So when seed is matured, right, seed dehydrate at the late stage of maturation. So biji benih biasanya akan dehydrate. Dehydrate, hilang air, right? Until the content of water is just up to 5 to 15% of its weight sahaja. Alright. So the embryo is surrounded by the food supply, either cotyledon or endosperm, or both. Depends on type of plant. Kalau monocot, usually we refer to endosperm. Kalau dicot, we refer to the cotyledon biasanya. Okay, right. And at the late maturation, embryo become dormant. Remember, dormant means it will wait until the time that it will germinate. That's why biji benih biasanya dia akan ringan-ringan lah. Dia akan kehilangan air. Okay, right. Dormancy in seed. So dormancy in seed is a condition whereby the growth and the metabolism of the seed is ceased or stop. Dormancy, dia dormant, dia tak buat apa-apa. Growth and metabolism will stop before it starts to germinate. So this is the characteristic of a mature seed. Dia akan dormant. Right, so dia akan kehilangan air and then dia akan stay dormant. Okay, right. Okay, next what happened? Okay, the embryo is differentiated into 3 parts. Okay, embryo dibahagi kepada 3 bahagian. First, radicle. Okay, which one is radicle? Ni, this is radicle. which will develop into the embryonic root. Okay. Next is plumule. Which one is plumule? This is plumule. Right. Plumule consists of epicotyle. Okay. Tak ada kat sini, nanti boleh tunjuk kemudian. Epicotyle, young leaves, shoot, and apical meristem. Shoot, apical meristem, and 1 to 2 cotyledons that will become the seed leaves. So this is cotyledon. Cotyledon ni jadi apa besok? Dia akan jadi the leaves, the seed leaves. Okay, right. So pernah kalau soalan MCQ, what are the component of embryo? So embryo comprises of radicle yang akan besok jadi akar, plumule yang besoknya akan jadi young leaves, shoot, epicotyle, okay, and then seed leaves, 1 or 2 cotyledon. In this case, you can see there is 2 cotyledons. Okay, right. And this is the remains of endosperm, right? And the embryo is actually covered with coat that we call as testa. Okay, bayangkan macam biji kacang hijau. So kulit dia yang hijau tu adalah testa. Okay. Okay. Next part is below where the cotyledon are attached, bahagian bawah cotyledon, below where cotyledon are attached, okay, the embryonic axis is called as hypocotyl. Hypocotyl. So this is hypocotyl. Okay, it is below the kita nak kata below where cotyledon are attached. Bahagian bawah cotyledon attached. So that is hypocotyl. Okay, hypocotyl terminates in the radicle. So maknanya this is radicle. Radicle besok akan jadi root. Okay, hypocotyl terminates in radicle. So when roots is developed, hypocotyl nanti akan terminate lah macam tu. Okay. The portion of embryonic axis above where the cotyledon are attached, okay, atas daripada cotyledon, and below the first pair of miniature leaf, this is miniature leaves, okay, is known as epicotyl. So below this is miniature leaf, below the miniature leaf is epicotyl. Right, so epicotyl, hypocotyl ni jadi apa? Nanti dia berkaitan. Hypocotyl nanti kita tengok, right. So ada yang jadi akan bahagian support, bahagian akar, and some akan become the shoot. Okay, the epicotyl. Right, so that is part of epicotyl and hypocotyl. Okay, right. So this is garden bean. So ingat, garden bean is unicorn. Right, maize or corn kernel is monocot. Okay, so for monocot, you can see that this is epicotyl. Ingat, epicotyl dekat dengan miniature leaves. Okay, and down here, right, before the radicle is the hypocotyl. Okay. Next, kita pergi kat sini. The structure of monocot seed and udicot seed. Okay, so monocot seed, okay, they have specialized cotyledon that we call as scutellum. So this is scutellum. Kita panggil dia scutellum. So this is the cotyledon of monocot, scutellum. To compare with the cotyledon of udicot. So udicot kita panggil cotyledon macam biasa. But for monocot we call it as scutellum. Alright. Okay. Next is they have a structure that we call as coleoptile. This is for monocot. Coleoptile. This one. Okay. What is the function of coleoptile? Coleoptile covers the young shoot. Right. It covers the young shoot. While the next one is coleorhiza. Coleorhiza is here. Right, that covers the young root. Okay, so this name, okay, or term is meant for monocot. It is meant for monocot. Okay, Dia ada apa nama? We call as structure, specialized structure and name. Okay. So for Unicode, we have macam biasalah kita ada epicotyl, the hypocotyl, radical, okay, ada cotyledons, okay. Okay, the fleshy cotyledon store food absorbed from the endosperm before the seed can germinate. Right. So germination of seed. Okay, remember at the late maturation of seed, the seed is actually losing water, right? Or dehydrate. Okay. So now and become dormancy. So when seeds start to germinate, right? So there are a few things that happen to induce the process of germination. Okay. Dormancy increases the chances that germination can occur at the time and place that is most advantageous for the seedlings untuk proses percambahan. Okay. Breaking dormancy and stimulation of germination requires certain environmental conditions like water, enough water, suitable temperature, light, and growth factors or growth promoters. And duration of dormancy is varies from one species to another species. Maksudnya, okay, excuse me, I need to plug in the battery. Okay, sorry. Right. So, seed will germinate at proper time and place. Okay. And before seed can germinate, remember the seed will stay dormant, but the duration of dormancy is differ from one species to another species. Okay. So the next question is, what are the mechanisms? So what is the biochemical mechanism for seed germination? For seed germination, right? So we will see the example of cereal grain. So cereal grain here is referring to the monocot lah. Kita refer to monocot. Okay. So this is the mechanism for germination in monocot. Example, cereal grain. So you can see that here is the endosperm, right? So we have cotyledon di sini, ataupun kita panggil scutellum. Right, the plumule and also the radical. Okay. Okay, so how the process or mechanism of seed germination is initiated. Okay, so first, right, it started with the process of inhibition. So ingat mechanism kita belajar ni adalah mechanism of germination in monocot, right? So it started with the process of imbibition. Remember what happened to the seed at the late maturation? Dia dehydrate, kehilangan air, kan? So untuk dia germinate, macam contoh kita nak tanam toge. Kita buat apa dengan kacang hijau tu? Kita rendam dalam air kan? Kenapa kita rendam dalam air? Right? So rendam dalam air is to induce the process of imbibition. Okay? So imbibition is the uptake of water due to low water potential of the dried seed. So the seed will take in water by the process of imbibition. Right? So the cell ataupun the seed then will swell. So the seed coat expand and rupture. So kacang hijau tu pecahlah dia punya seed coat yang warna hijau tu. Right? So this is happen after the process of imbibition. So seed expand and rupture the coat, the seed coat. Right. Next, cellular respiration started. Okay, what happened next? Embryo release GA, right? Gibberellin hormone. Okay, embryo will release gibberellin into the endosperm. Okay, right. Let's look at the picture. This is endosperm. Okay, so first it started with the process of imbibition. Right, the seed takes in water, right, and induce the release of gibberellin. Okay, so gibberellin is released into the endosperm, and gibberellin acts on aileron layer. So in other words, what is the target cell of gibberellin? The target layer or the target cell of gibberellin is aileron layer. So where is aileron layer? Ni, yang ada bintik-bintik ni aileron. So you can see gibberellin is acting at the aileron layer. So apa dia buat as it acts on the aileron layer? So gibberellin acts on aileron layer by stimulating aileron layer to synthesize digestive enzyme. Stimulate aileron to synthesize digestive enzyme. Enzyme hydrolyze the stored food inside the endosperm into a soluble molecule or monomers. Okay, let's see here. The aileron layer induce the secretion of hydrolytic enzyme or digestive enzyme, which, for example, amylase will break down starch into simple sugar, right? So inilah kita panggil sebagai enzyme hydrolyze the stored food in the endosperm into a soluble monomer. Okay, right. So next example, despite amylase that hydrolyze starch into disaccharide or into monosaccharide or sugar or glucose, protease, right, break down protein into amino acids. Lipase break down lipid into fatty acid and glycerol. So this is what happened when aileron layer stimulate the release of digestive enzyme. Okay, right. So what's next? So nutrient yang tadi tu, all the nutrients like amino acid ke, glucose ke, fatty acid, glycerol ke, will be mobilized by the enzyme and absorbed by the scutellum. So in other words, right? So sugar, for example, will be absorbed by the specialized cotyledon that we call as scutellum. Okay, right. Okay. So next, what happened? The nutrient also is transferred to the embryo, right? And nutrient is used by the embryo for growth, to synthesis polymers, and also some of the nutrient will be translocated to plumose and radicle. Okay, so you can see here some of the nutrient is transferred to the plumose and to the radicle, because plumose later on will develop into shoot and radicle later will develop into young. Okay. Okay. So the first organ to emerge from the germination of seed is radicle. So organ pertama yang akan keluar during the germination is the radicle. That's why yang keluar, kita tengok kalau kacang hijau tu yang keluar naik dulu tu akar dia. Okay, right. Okay, again the picture so of the scutellum. So nutrient is here from the endosperm, is transported to the scutellum and then mobilized to the plumule and also radicle. Okay. Okay. Right. Next, last LO for this chapter is growth of seedlings. So seedlings mean percambahan. Okay, percambahan. So there are two types of seedling. Ada dua cara biji benih bercambah. Dua cara. Okay. So growth of seedling. Germinating seed continues under metabolic changes and culminating into a seedling. There are two types of seedlings grow. Ada dua jenis. Two types of seedling growth. Okay. The first one is apigeal germination. So two types. Number one is apigeal germination. And number two is hypogeal germination. Okay, two types of seedling, apigeal germination and hypogeal germination. Okay, so you must be able to differentiate hypogel and epigel germination. Okay, so let's start with epigel germination. Okay, epigel germination usually happen in sunflower, long beans, garden beans, which are referring to many eurycod, right? So most of eurycod undergo epigel germination. Okay, right. What happened during epigel germination? During epigel germination, hypocotyl. Ingat balik, hypocotyl tu apa? Hypocotyl is area below cotyledon. Bahagian yang di bawah daripada cotyledon. Okay, area below cotyledon. So, in epigel germination, hypocotyl will elongate. Yang akan memanjang ialah hypocotyl. Okay, so what happened to epicotyl? So next, first is epicotyl. In epigel germination, hypocotyl germinate. Elongate. Okay, it will push as hypocotyl elongate. It will push epicotyl and cotyledon above the soil surface. Dia akan tolak epicotyl and cotyledon above the soil surface. The exposed epicotyl spread it first through leaf and expand, become green and making food by photosynthetic, right? So since cotyledon is also pushed above the soil, cotyledon become photosynthetic as well. The seed coat emerge from the soil. Seed coat akan keluar daripada tanah. Maksudnya akan nampaklah dekat atas tanah. That is epigel germination. Okay, tak nampak kan macam mana kan? Kita tengok gambar dulu. Right. So this is common garden beans. So remember this is eurycod. Right. So what happened in epigel germination? In epigel germination, the one that will be elongate is hypocotyl. Ni hypocotyl. Okay. Hypocotyl kat sini, radical kat bawah. Right. Okay. Hypocotyl elongate. You just imagine benda ni memanjang. Nampak ni? Hypocotyl elongate. This is radical. Pada radical. Okay. When hypocotyl elongate, you can see that what happened? It push the cotyledon and also epicotyl. Dia tolak, dia bawa sekali this cotyledon as well as epicotyl above the soil. Bawa naik ke surface. Right? And then emerge the true leaves. Epicotyl emerge its true leaves. Right? And what happened to cotyledon? Cotyledon become photosynthetic as well. Okay. So, madam, tak nampak pun mana satu epicotyl, mana satu hypocotyl. Okay, tengok sini. Ini cotyledon. Area below cotyledon is hypocotyl. Okay. Area above cotyledon is epicotyl. So you can see when hypocotyl elongate, dia akan tolak sekali both epicotyl and cotyledon above the soil surface. Right, which makes cotyledon become photosynthetic. That's why you can see the seed coat also emerge. Seed coat emerge on the soil surface. So this is kind of epigel germination. Okay, right. Okay, madam buat simple conclusion kat sini. Okay, ingat in epigel germination, the one that is elongate is hypocotyl. Elongate. Okay, dia terbalik. So in epigel germination, the one that is elongate is hypocotyl. Okay, right. Okay, next. Okay, remember cotyledon become photosynthetic. The seed coat above emerge above the soil. Okay, right. Okay. In kejap lagi boleh tunjuk video tu. In hypogel germination pula, right? Hypogel germination usually occurred in many monocots like wheat, rice, maize, and also grass. Okay. In hypogel germination, the one that is elongate is epicotyl. Okay, area above cotyledon. Right? So yang memanjang adalah epicotyl. So apa jadi dengan cotyledon? Right? So the one that is elongate is epicotyl. So thus it will pushes coleoptile above the soil surface. Okay, ingat tadi balik. Coleoptile ni function dia apa? Cover the young shoot. Okay. Covers young shoot. Okay. Alright. Okay. So as epicotyl elongate, it pushes coleoptile above the soil surface. So the shoot tip grows and break through the coleoptile tip upon erosion. What happened to cotyledon? Cotyledon remain underground. Sebab yang memanjang adalah epicotyl. Epicotyl tu bahagian atas daripada cotyledon. So dia tinggallah cotyledon tu dalam tanah. Okay. So the cotyledon remain underground. Cotyledon does not become photosynthetic, and the seed coat also remain inside the soil. Okay, that is hypogel germination. Okay, kita tengok gambar kejap. Right. Example here, maize, monocot. Right, so you can see here, during germination, siapa yang memanjang? Yang memanjang ialah epicotyl. Epicotyl elongate. So when epicotyl elongate, it pushes the coleoptile above the soil surface. And what happened to cotyledon? Cotyledon remain in the soil or remain underground. So is cotyledon become photosynthetic? No, cotyledon does not become photosynthetic. Right? So the seed coat also still ataupun the seed coat also remain in the soil. Okay? Okay, so short notes. Okay, for the hypogel germination, in hypogel germination, the key is epicotyl elongate. Epicotyl elongates. Right? So it will cause the cotyledon to remain in the soil and not photosynthetic. Right? Okay. Jom tengok video ni kejap. This is one here. Tengok, is input inside the soil. Right? Or you can and what is right? So macam this is the color. This on the side. Okay, Okay, nampak tak? The difference between hypogel germination and epigel germination. Can you see the difference?
Yes, madam.
InsyaAllah. Okay, right. So ingat the key given to you. Epigel germination, hypocotyl elongate. In hypogel germination, epicotyl elongates. Okay. Right. Okay, again, you can see this is for monocotyledon, right? Epicotyl elongate. So it involved in hypogel germination. Right, so you can see epicotyl elongates. It pushes coleoptile above the soil surface, right? The what you call it, seed coat, cotyledon and seed coat remain underground or in the soil. Okay, whereby here this is dicotyledon, right, or udicot plant. You can see what happened. It involved in epigel germination. Epigel germination. Okay, so in epigel germination, the one that is elongate is hypocotyl. So you can see when hypocotyl elongate, it pushes the cotyledon, right? And also the what you call it, coleoptile, ataupun it pushes the plumule to the soil surface. Okay, it pushes together. And then cotyledon become photosynthetic. Okay. Right. So that's all. Okay. Mark the end of chapter 9. Habis lah chapter 9. Right. Any question? Question?
No, madam.
Tak ada. Okay, hopefully okay. And you can, in case want to watch again the what you call it, the class recording, right? So I will put it in YouTube, inshallah. Okay. So